Okay, guys, today we are going to explain the process of meiosis. Let's watch together the video and discuss it. I just want to make sure that the sound is working. Just 1 minute, guys, please. Yes. Okay, listen carefully so you can understand and we can discuss every single point.
Not look exactly like their parents, or why some siblings look
Sorry, I want to start it from the beginning.
so different from each other? The answer lies.
All right.
Have you ever wondered why children do not look exactly like their parents, or why some siblings look so different from each other? The answer lies in the process of meiosis, which generates the gametes for reproduction.
So, this is a very important point. Maybe it comes
Yes,
to your
miss.
mind. Yes, Muhammad.
بس حكيت يس مس اني موجود.
Sorry?
مس رحت اجيب شغله فاجيت حكيت يس على اساس اني موجود بس.
No problem. Okay, maybe there's a question that come into your mind that how the siblings from the same parent are different. And you and your sisters and brothers, you are similar but not identical. You have some variation between yourselves. And even if the mother and the father are the same, but you still have differences. Murad, you want to ask any question?
No, miss. كنت أجاوب إذا سألت السؤال.
Yes, answer it. Why? Why there is a variation between the siblings?
Because of meiosis.
Yes, because there is a process of division for the germ cells, which is called meiosis.
Meiosis is the process by which haploid cells are produced from a diploid cell. For this to occur, the chromosomes must be correctly sorted and distributed in a manner to create genetically unique cells with half the number of chromosomes as the original cell. Meiosis occurs in special cells called germ cells within the gonads of males and females. For the successful reduction in the number of chromosomes to occur in the new haploid daughter cells, 2 rounds of divisions are necessary. These divisions are termed meiosis I and meiosis II. Although the processes of mitosis and meiosis look similar, there are a few major differences.
Okay, now you have to keep in your mind, in mitosis, a division that happens to the body cells, and meiosis is a cell division for germ cells. All the germ cells are cells that located in the reproductive organs of males and females. Both of these processes start with a diploid cell. Diploid cell يعني a cell that have a whole number of chromosomes. يعني germ cell before it differentiate to sperm or egg كانت diploid. When body cell also it's a diploid. الgerm cell كانت 46 chromosomes. زي body cells كانت 46 chromosomes. Now, during mitosis, when the body cells divide, it produces 2 daughter cells with the same number of the parent cell, which is 2 diploid cells, okay, with same number, 46, 46 each one. While in meiosis, high diploid germ cell, when it divides, it goes through 2 stages, meiosis 1 and meiosis 2, to produce 4 haploids. That means that each one has half the number of the chromosomes. Okay?
Obvious difference is that mitosis results in 2 diploid daughter cells, and meiosis results in 4 haploid cells. As in mitosis,
Okay, meiosis results in haploid cells, but the original parent كانت diploid. بالميتوسيس عندي 2 diploid cells والoriginal اللي هي الparent كانت diploid. So both of these processes started with a diploid cell. واحدة germ بالميوسيس والثانية body cell بالميتوسيس. This is very important.
Meiosis begins after a cell has successfully completed the G1, S, and G2 stages of interphase. During the S phase of interphase, the DNA is replicated.
We learned that during the cell cycle, the cell goes through different stages. Let's start with the interphase, اللي هو preparing, by growing, and copying of the DNA. تمام?
Producing 2 copies of each chromosome called sister chromatids. The paired centrioles in the cytoplasm duplicate and begin extending microtubules that will form the meiotic spindle. Sister chromatids remain attached at the centromere and condense as the cell enters prophase 1 of meiosis. Up to this point, the cell looks similar to mitosis, but 2 events occur in meiosis that do not occur in mitosis.
Okay. Is my voice clear, guys? Hello, is my voice clear?
No.
Miss, it's
No,
like
miss.
a little bit.
Miss,
Yes.
it's glitching.
Yes.
Now, just now?
It's glitching.
Just now?
Yes, now.
Okay.
Oh,
We will repeat this part. Okay. Now here, and we start with meiosis. Okay. Meiosis, it has 2 stages. In the meiosis 1 and meiosis 2. Now it's clear. My voice is clear, guys. Now.
Yes.
Okay.
Yes. Yes.
هلا بميوسيس
yes, miss, yes.
بميوسيس 1 كانت السيل الجيرم سيل فيها 46 كروموسوم. اوكي. هلا what will happen احنا we learned انه during the cell cycle after interphase during interphase راح يصير عندي copying للدي ان اي. تمام؟ وكل دي ان اي حيكون عنده 2 sister chromatids which joined in the centromere. Right? تمام. Clear till now? تمام. هلا here
Yes.
the germ cell it have 46 chromosomes. عندكم من each chromosome you have 2 pairs, 2 homologous chromosomes, one from the mother and one from the father. صح؟ From each one chromosome you have 2 homologous. Okay? Let's
2 events occur in meiosis that do not occur in mitosis and lead to genetic diversity. The first event occurs during prophase 1, when homologous pairs of sister chromatids lie side by side in a process called synapsis, forming a tetrad.
هلا this process هلا بالmitosis this process doesn't happen. هلا the phases are the same, prophase, metaphase, anaphase, and telophase. They are the same. But let's see what is the difference in each phase between mitosis and meiosis. هلا بprophase 1 بالmitosis this is not happening. In prophase 1 بالmeiosis 2 homologous chromosomes will join together, not join, come near to each other and make this structure اللي هو tetrad. 2 homologous, one from the mother, let's say the purple from the mother and the blue one from the father. Okay, they will come near to each other and make this structure اللي هو tetrad. We call it tetrad. I don't know why Baha cannot admit. Baha, do you have a problem, Baha? I don't know what's wrong. Can you hear me, guys? Guys,
Miss,
can
yes,
you
we
hear?
can hear you.
Yes, miss, yes.
I don't know. Baha has a problem. I don't know what's anyway. All right. So this doesn't happen in mitosis. With mitosis, just the chromosome will copy. Will have two sister chromatids. تمام? But the 2 homologous chromosomes will not come near to each other like in meiosis. هذا هو الديفرنس. إذن the first thing that is different between mitosis and meiosis is forming the tetrad structure here, where the 2 homologous chromosomes will come near to each other.
Or bivalent. The homologous chromosomes share similar but not necessarily identical genes. Once this
As we learned in the previous lesson, in the homologous chromosomes, they are similar but not identical. تمام? They are not identical. أما sister chromatids of each chromosome, they are identical. Okay? Did you get this point? Do you understand it?
Yes, miss.
Okay, perfect. إذا هون two homologous chromosomes, they are similar but not identical. Look what will happen here.
Structure is formed, the second event, called crossing over, can occur. During crossing over, a physical exchange between chromosomes.
Now, while they are near to each other, these two homologous chromosomes, crossing over process may happen between them. And what this means, parts of the chromosomes will be, look here what will happen.
Segments of non-sister. During crossing over, a physical exchange between chromosome segments of non-sister chromatids occurs.
يعني هدول two sister chromatids اللي هم البurple والblue are two sister chromatids. During crossing over, while these two chromosomes are near to each other, there is exchange in parts. شايفين؟ صار عندي exchanging between non-sister chromatids. يعني not between the two sister chromatids, between one sister with the other chromosome sister chromatids. شايفين؟ And this will make variation in the genes. صار عندي اختلاف هون بالجين from this process. And this process doesn't happen in mitosis, guys, right? This doesn't happen in mitosis. تمام.
Increasing genetic
الصوت
diversity.
كثير عم بيقطع.
Okay, guys, I will repeat. Okay, now you can hear me?
Yes, yes. Now, yes.
إذن, I will repeat what I said. During prophase, okay, after the two homologous chromosomes make this tetrad structure, they become near to each other, crossover process will happen. بالميتوسيس this doesn't happen لأنه أصلا الهومولوجous they don't attach to each other. And what is the meaning of crossing over? That there are exchange between non-sister chromatids. يعني this chromatid will exchange some parts with the other chromosome's chromatid. Okay?
Prophase 1 concludes with the fragmentation of the nuclear envelope as the duplicated centriole pairs move to opposite.
Like mitosis, exactly. The nuclear envelope, which is enclosed the nucleus, will disappear, and the centrioles will move to opposite sides of the cell.
Poles of the cell. As they move, the centrioles extend spindle fibers, forming the meiotic spindle. In prometaphase 1, with the paired centrioles in place, the meiotic spindle is fully formed. The sister chromatids attach to the spindle fibers by their kinetochores. Here, another key difference between mitosis and meiosis occurs. Because of synapsis and crossing over in meiosis, homologous chromosomes remain aligned, so that a pair of sister chromatids is attached to only one pole by the kinetochore microtubules. In mitosis, each sister chromatid is attached to a spindle fiber. The fibers originate from opposite poles.
شايفين the difference? هلا بالميتوسيس each chromatid will be connected to a spindle fiber. But in meiosis, the whole chromosome from each two homologous chromosomes will be attached to the spindle fiber.
During metaphase 1, bivalents randomly align.
Prophase 1 finished. Metaphase 1, like mitosis, they will be aligned in the middle of the cell. But what will be
along the metaphase plate? Due to independent assortment, this alignment is random and adds to genetic diversity. In anaphase 1, the homologous chromosomes separate and move toward opposite poles.
Look, what is moving opposite towards each pole? The whole
بس الصوت كثير عم بيقطع كثير.
I don't know. I have the internet here is good. I'm not sure what's going on. Then I'll try to check here. Just 1 minute, guys, please.
There is no sound. صوتك كثير عم بيقطع كثير.
And now it is also the same.
هسه لا بس طول الوقت وانت مشغل اليوتيوب ولا كان مبين صوت هيك.
Okay, I will repeat from here. From here?
Okay.
From here?
Yes.
Okay, yeah, we will repeat.
Physical exchange between chromosome segments
احنا قلنا during the prophase هدول هيدا I will start from here. Listen,
lie side by side in a process called synapsis, forming a
So during prophase, the homologous chromosomes will come side by side in a process. This process is called synapses, and they form tetrad structure. Okay, clear now, عبد الرحمن?
ما فهمت.
Again, is it clear? My voice, is my voice clear?
Yes,
نعم
yes.
صوتك واضح.
تمام. Okay, here يا عبد الرحمن and everyone, during a prophase, after the DNA copied, what will happen بالmeiosis 1? The two homologous chromosomes will come side by side in a process called synapses and form a tetrad structure like this.
diversity. The first event occurs during prophase 1, when homologous pairs of sister chromatids lie side by side in a process called synapsis, forming a tetrad or bivalent.
شايف عبد الرحمن? They lie side by side, and this process called synapses and form a tetrad or bivalent. This structure called tetrad. They are not attached. They lie side by side only. Okay? Clear عبد الرحمن?
Okay.
تمام.
Yes, yes.
Homologous chromosomes share similar but not necessarily identical genes. Once this structure is formed, the second event, called crossing over,
But while they are lying side by side, yes. Yes. Any question?
Yes, yes. Miss هسه these crossing over cross face one, these things, where are they located in the body?
In the cell يا مس. كل cell. بالcells اللي هم الgerm cells. الgerm cells that will become either an egg or sperm. Okay? بس لما يصير عندي division للgerm cells that will become later egg or sperm. يعني هلا في الميل الgerm cells اللي موجودين في reproductive system, when they divide, they will divide in this process اللي هي meiosis. رح يصير عندي this crossing over inside each cell and they will produce sperms. أما بالفيميل this will produce eggs. واضح عبد الرحمن؟
بس صوتك راح.
Okay.
Hello.
Hello, yes, عبد الرحمن. Can you hear me now?
Miss.
Yes, can you hear me? Hello, can you hear me, guys?
ما في صوت.
أبدا؟
Hello.
Yes, just 1 minute. I'm trying to fix it here, guys. Just 1 minute, حبايبي. Wait. Hello, can you hear me now, guys?
Hello. صوتك اختفى فجأة لأنه.
Now it's clear. Is it clear
Yes,
now?
your
طيب.
clear.
Okay, so هلا هاي كروسنج اوفر عبد الرحمن فهمت علي انه هلا هدول هاي البروسيس الميوسيس بتصير ديفيجن للجيرم سيلز اللي موجوده في الريبرودكتيف سيستم. بالميلز هدول افتر ميوسيس بصير عندي سبيرمز وبالفيميلز بصير عندي الايجز. اوكي؟ يعني هذه موجوده بالسيلز اوف ذا جيرم سيلز. طيب ماشي عبد الرحمن مفهوم؟
ماشي مفهوم.
تمام؟ هلا الكروسنج اوفر هذا معناته صار عندي انا exchanging تبادل بالجينز between these two homologous chromosomes. See what happened?
Can occur. During crossing
Yes.
over, a physical exchange between chromosome segments of non-sister chromatids occurs, increasing genetic diversity. Prophase 1
بيعمل عندي genetic diversity في اختلاف بالجينات. Okay, that cause different siblings. لأنه صار عندي اختلاف، صار تبادل بالجينات between the chromosomes، فهذا will make changes أو differentiation between siblings, variation between siblings اللي هم ال brothers and sisters، لأنه بطل الكروموسوم نفسه، صار عندي تبادل بالجين. شايفين؟
1 concludes with the fragmentation of the nuclear envelope as the duplicated centriole pairs move to opposite poles of the cell. As they move, the centrioles extend spindle fibers, forming the meiotic spindle. In prometaphase 1, with the paired centrioles in place, the meiotic spindle is fully formed. The sister chromatids attach to the spindle fibers by their kinetochores. Here, another key difference between mitosis and meiosis occurs. Because of synapsis and crossing over in meiosis, homologous chromosomes remain aligned, so that a pair of sister chromatids is attached to only one pole by the kinetochore microtubules. In mitosis, each sister chromatid is attached to a spindle fiber. The fibers originate from opposite poles. During
يعني هون بالميتافيز 1 بالميوسيس the whole homologous chromosome, the two chromatids will be attached to the microtubules. هناك بالميتوسيس each sister chromatids will be attached. يعني هون عندنا the whole chromosome will be attached. بالميتوسيس لا, each sister chromatid will be attached.
Metaphase 1, bivalents randomly align along the metaphase plate. Due to independent assortment, this alignment is random and adds to genetic diversity. In anaphase 1, the homologous chromosomes separate and move toward opposite poles. Meiosis.
Look at the chromosomes here. In anaphase 1, they start to divide and separate to different poles, different sides of the cell. Well, look how the homologous chromosome look like. They have mixing genes here. They are not like the original one. صح؟ صار في عندي variation, في اختلاف. شايفين؟ From the crossing over.
Meiosis 1 ends with telophase 1, when the chromosomes decondense and the nuclear envelope reforms. Cytokinesis separates the cytoplasmic material, and the two daughter cells are separated by a cleavage furrow.
Now here, هذا meiosis 1 ends at this stage. بصير عندي cytokinesis and nuclear envelope will form again, and they will have in meiosis 1 two cells.
Since the final product of meiosis 1 is two haploid cells, meiosis 2 begins without the chromosomes going through another round.
Here, at the end of meiosis 1, we have two haploid cells. ليش؟ لأنه two homologous chromosomes, half of them go to one cell and the other half go to the other cell. بطلوا 46, صاروا haploid. All right?
Of DNA replication. Centrioles again duplicate and begin moving to opposite poles of each cell. In prophase 2, the sister chromatids condense while the spindle starts to form as the nuclear envelope disappears. In prometaphase 2, the sister chromatids attach to the spindle by kinetochore microtubule.
هلا what happening here is like mitosis, بالضبط زي الميتوسيس عم بتصير. أنا صار عندي الهومولوجous chromosomes they separated to different cells في meiosis 1. في meiosis 2, the prophase will start like mitosis. They will duplicate it, copies, and they will arrange themselves in the middle, look.
With sister chromatids attached to opposite poles. The spindle aligns the sister chromatids along the metaphase plate during metaphase 2. During anaphase 2, sister chromatids.
What happened here? The sister chromatids were separated, not the whole chromosomes. شايفين زي الميتوسيس بالضبط. في الميتوسيس, this what happening. The two sister chromatids will be separated.
Separate and individual chromosomes move toward the poles. The entire process ends with telophase 2, as the chromosomes decondense and the nuclear envelope reforms.
So each cell will result two cells, and at the end we will have 4 cells.
Cytokinesis occurs, and cleavage furrows separate the two daughter cells into 4 haploid daughter cells. The haploid daughter cells will specialize into gametes, either sperm or egg. These fuse in fertilization to form a zygote, which will grow into a child. The child receives half its chromosomes from its mother and half from its father. Because men and women produce millions of gametes, and the selection of gametes in fertilization is random, this contributes to genetic diversity. This explains why a child isn't identical to either parent.
So can anyone answer the question why the siblings are not identical? There is variations between them. Yes, Murad.
Because meiosis produce millions of gametes, which will then be chosen randomly in
Randomly.
fertilization.
برافو. During the division of the germ cells هدول الجميع رح يصير فيهم crossing over، فراح يكون عندي homologous chromosomes will not be identical like the parent cell. صح؟ فهذا الفاريشن بصير عندي randomly، بصير عندي مثلا one egg with a sperm will produce different variation from other egg and sperm from two same parents. Okay? Okay.
But why aren't siblings identical? The diversity comes from several sources. In prophase 1, non-sister chromatids can exchange DNA through crossing over either parent. But why aren't siblings identical? The diversity comes from several sources. In prophase 1, non-sister chromatids can exchange DNA through crossing over, increasing the genetic diversity of individual chromatids. In addition to crossing over, in metaphase 1, the pairs of homologous chromosomes align independently along the metaphase plate and sort independently into the daughter cells. This process, called independent assortment, produces 4 genetically distinct haploid gametes. As the total number of chromosomes increases in an organism, the number of genetically distinct gametes increases by 2 to the nth power. This means an organism with n equals 3 can produce 8 unique gametes. For humans where n equals 23, there are 2 to the 23rd power unique gametes formed, or over 1 million different possible combinations.
Okay. Anyone has any question about this video, guys? Anyone has any question, please?
Miss,
Yes,
can you please send it to us in the group?
of course.
Because
I will
watch
send
it.
video, the link of the video during the class, and I will send you the video also. Okay? Of course.
Okay.
I want to show you another thing. There is a very important thing I want to remind you about, which is this thing. Let's show you. Okay, this is from your book. Now, to differentiate, you have to differentiate between 2 important things. The chromosomes. Yes. Yes, miss.
لا ما في شيء ما في شيء.
Okay. You have to understand this carefully. The difference between homologous chromosomes and sister chromatids. You should not mix up with these 2 terms. Homologous chromosomes, what are they? They are very similar but not identical. In each cell in your body, body cell, in each cell, حتى germ cells before meiosis, each cell in your body عندها homologous chromosomes that are similar, okay, and carry the same genes but not the same version of the gene. But they are not copies of each other. In homologous, they are not copies. They are similar, but they are not copies. Just 1 minute, I want to see the chat here. Okay. This one not today, Yazan. I will send it for you tomorrow, maybe, inshallah. Okay, when we finish the lesson. So homologous chromosomes are chromosomes that are similar, have the same genes, but they are not identical. And they are not copies, yani. Okay? You get one of them from the mother and one of them from the father. هدول هم الهومولوجous chromosomes. Okay? Now, the sister chromatids, okay, each half, when the chromosome duplicated, each half is called chromatid. لما يصير عندي duplication للكروموسوم نفسه، هذا هو كل half منه بصير اسمه chromatid. Okay, إذا each half of a duplicated chromosome called the chromatid. Together, the two chromatids are called sister chromatids. هلا sister chromatids, are they identical or not? What do you think? Do you think the sister chromatids are identical? عبد الرحمن?
Yes.
Yes. Not like the homologous chromosome, one from mother, one from the father. They are similar, have same genes, but not identical. أما sister chromatids, I want to use this. هيهم هدول. Just 1 second. هدول two sister chromatids. When they will form هدول? When the chromosome duplicated, copied during the cell cycle, the chromosome make copy and make two sister chromatids. هدول two sister chromatids are identical because it's a copy of from the original chromosome. أما الهومولوجous, no, they are similar but not identical. فبالميتوسيس guys, بالميتوسيس what's happening really إنه هو separation of the chromatid. بالميوسيس 1 يصير عندي separation between homologous chromosomes. Clear, more clear now. حدا عنده سؤال؟
No.
حدا عنده no question? طيب. I want to show you another video also to see the difference again to make it more clear. Okay, just 1 minute. All right, we'll start.
Cell cycle is one of the most breathtaking processes that helps the cell to not only grow but also divide. We know that a cell cycle has two major
إذا
phases.
الcell cycle برضه I keep on reminding you هي cell cycle during where the cell grow and divide. تمام.
One helps in the preparation of the cell for the division. Can you name the preparatory phase of the cell cycle? That's the right answer. It's called the interphase. And moving to the other phase, it carries out the scission of the cell. And what is that phase called? Correct. It's the M phase, where M stands for either mitosis or meiosis.
يعني
And how to
the cell cycle on الانترفيز هي the preparation stage. Then will come the division stage, which is either mitosis or meiosis. إذا كانت body cell mitosis, إذا germ cell meiosis.
does the cell decide whether to undergo mitosis or meiosis? Well, that depends on various factors and the type of cell undergoing division. Keep your eyes glued to the video to spot the differences between mitosis and meiosis. Let me help you with the first difference between the two processes. Mitosis occurs in somatic or body cells, while meiosis occurs exclusively in the reproductive cells.
The germ.
That means mitosis, occurring irrespective of whether the individual is a male or a female, gives rise to somatic cells, which are exact copies of the parent cells. On the other hand, meiosis occurs in males and females, giving rise to haploid sperm and egg cells respectively. The process of meiosis, however, has different names in both the sexes. Yes, due to the different cells produced as an end result, meiosis is also referred to as oogenesis, that is the process of ovum production in females, and spermatogenesis, that is the sperm production process in males. Another major difference is that mitosis is used as a mode of asexual reproduction in lower organisms, while meiosis helps in forming gametes for sexual reproduction. Now, let's move ahead with the next difference. Have a look at these images. Observing this image, we can easily notice the most basic differences between the two. Let's begin noting down each difference individually. The first on the list is that mitosis has only one cell division, while meiosis involves two successive cell divisions. That simply means mitosis is a single-step division, while meiosis is a two-step division. And this results in meiosis having two phases, namely meiosis 1 and meiosis 2. Can you note down the second difference in the list? Correct. Mitosis produces 2 daughter cells, while meiosis produces 4 daughter cells. And is there something remarkable that we haven't discussed yet? That's right. Now let's move on to the next major difference. This difference is based on the chromosomal number of the daughter cells. In mitosis, the daughter cells are diploid with 2 sets of chromosomes, just like the parent cell, while during meiosis, the daughter cells produced are haploid with only 1 set of chromosome. These daughter cells are called gametes, have exactly half the number of chromosomes from the parent cells. The former difference also gives cue about one more difference between the two. What could that be? Well, the image clearly depicts that all the 4 daughter cells produced by meiosis are genetically different from each other, while the 2 daughter cells that are produced by mitosis are genetically similar to each other. Now let's move on to the next difference.
Okay, we'll stop here, guys. And tomorrow, inshallah, we'll continue. Any question before I leave? Any question, guys?
No, miss, but don't forget to send us the videos.
Yeah, I will send the 2 videos for you على الواتساب وعلى البلاتفورم. Okay?
Okay, thank you.
Yeah, take care, guys. Take care. See you.